Go shorty, it's my birthday. We gonna party like it's my birthday. All right. I'll stop. All right, so, and it was the clean version. So welcome to Virtual Lab 21, the Stomata Lab. I am going to go over the entire lab, start to finish, to help you guys at home so you can turn in and we can then proceed and finish Lab 21 this week. So as long as you're watching this video and filling in all the questions and data, then we should be all set. And yes, it is my birthday today, and I'm sad that you guys are not here to celebrate it with me, but we will make the best of it. Some of you have, everyone else in here should have all of the pages prior to this, where some of you still might need number 6. So if you haven't, make sure that you're reading and creating some hypotheses, and let me know if you need more help. But number 6 is the leaves of the holly tree were found to have different densities on the stomata. Remember, stomata help to maintain water balance and gas exchange. So they say explain the difference would be the advantage for the tree. So obviously, having increased stomata would increase photosynthesis. Therefore, that's what those 3 little dots mean, or therefore, more sunlight needed. So that means that if this is my plant here, okay, let's say that this is my windowsill and my plant's kind of hanging, that the leaves in the direct sunlight here would actually have more stomata than if I tested the amount of stomata on the leaves towards the center of my plant or my tree. So more light, more stomata, so they can do a higher rate of photosynthesis. Right, so there's your hypothesis. Would every species of plants have similar numbers of stomata per millimeter squared? And the answer to that would be no. Every species is different, just like every single one of us is different. A pretty pond lily that lives in water is going to have only some stomata on the top and actually have quite a bit to get rid of the extra water so it doesn't have a hypotonic solution, compared to our cactus plant here in the classroom that is going to have a little bit different amount of stomata to keep water in because it lives in the desert normally, okay? So making sure that we are answering those for number 6 and your hypothesis. Your hypothesis does have to be as an if-then. So if species live in a different environment, then they'll have a different number of stomata per millimeter squared. Okay, so make sure that you have that for that part. All right, I'm going to scroll through. For number 8, you had to, this is where the part where we're at in lab, you had to calculate the number of stomata on a rhododendron. So just as a quick little review for some of you, so I took a leaf off of my rhododendron, which stays green all year around, okay? And remember that I cannot just simply take this leaf and put it under my microscope because it is too thick, right? Light does not go through, okay? And so to be able to see any structures, I need to make an epidermal peel. So what I do is I take a little bit of fast drying nail polish, get out the extra, and I go ahead and paint about the size of a penny on the lower side of the leaf, because for this species, it has a really thick, waxy cuticle. So I want to make sure if I want to see all the stomata that I get to the bottom. After about a minute, okay, let it air dry super fast. I then take a piece of clear tape and I go on top of the leaf, make a quick little run through, and then I can pull my tape up. This will act like my cover slip, so you can already see that you can see the structures of the leaves. I will then carefully place it on my microscope slide and put it on my microscope, okay? So here I'm going to take you to the microscope. Okay. So here is my field of view under high power, and I'm going to count how many stomata I have, and then I'm going to move the field of view 3 times. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25. All right. So going back to my data table here, I'm going to have 25 stomata, okay? All right, so now I need a different field of view, so I'm gonna do this 3 times. So again, here I am. I'm going to move my stage clip. So I'm just gonna simply move my field of view a little bit to look at a different part of my sample. And I'm gonna count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36 is what I count for that one. Okay, so I'm going to write that down. And then I'm going to move it one more time because I want to look at 3. So I have multiple trials to make sure my data is more accurate. So I had 36 and 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Okay, so that one had 17. So if you're watching, you can fill in the same data. 36, 17. And then go ahead and calculate your own average. So you're going to add them all up, hit the equals, divide it by 3, and round to the nearest whole. Then you're going to take this number. This is in, okay, under high power. I want to know how many are in, okay, they're saying calculate how many stomata per millimeter. So I'm going to take this number that I calculated and divide it by 0.45 to get my number for number 9. So again, the question would be, would every species of plant have similar number of stomata? We're going to say no, because every species have adapted to different environments. Also, the different leaves even have different number of stomata depending where they are, in more direct sunlight or in less sunlight. So there's your question from there. For procedure 2, okay, the guard cells are responsible for opening and closing the stomata when water concentration is high. The guard cells will bulge. Remember, it's all dealing with osmotic pressure. When the water concentration is low, the stomata will close. Stoma are generally open when the plants are photosynthesizing. Will plants have more stoma open during the day than during the night? This is where I would like you guys to come up with what you think it is. Do you think they would have more open stomata in the night or the day? You just have to write night or day for number 10. So if you choose night or you choose day. Then for number 11, they want you to, in a complete sentence, have your hypothesis. Okay, so every great hypothesis for Regents biology should start with an if. So if the plant is in daylight, then it will have more open stomata because it can then do photosynthesis, which is needed for, it needs light for that reaction. Or you could say if stomata are in the dark, they would have more open stomata to get rid of extra water because of the dark reactions. You get to choose what your hypothesis is. I'll give you a second. You can pause, get your hypothesis in. All I care about is when I look at your lab, you have an if-then statement, and you tell me what do you think, in using your plant physiology, would there be more open stomata in the night or in the dark? So now, what I did do was I actually did this part for you guys. I took some leaves of my rhododendron and I actually put them in the dark, and then I set a bunch of my leaves in the sunlight. And I did the same thing, okay? I did my epidermal peel of 3 different samples in the light and in the dark, and I got a percentage of how many were open. So I'm gonna bring you back over here to my microscope. Okay, I know it's a little bit harder to see here. If there is, like you can see this one, there's no dark space in the inside, that means that stomata is wide open. If there is, it's nice and dark and there's a line through it, like this one over here, that means that stomata is closed. And then I could calculate a percentage of how many are open by doing the number of open divided by the total number, and that would give me my percentage. So my numbers, okay, for my data table, which you need to be filling in, is for light, I got 85, these are percentage, 85% on my first slide of open, 83% on my second slide of open, and 84% on the light. So again, zooming in a little bit here so you can see my data table. Light, I got 85, 83, and 84. You then need to calculate an average, so you do got to do a little bit of work for this lab. So I'm giving you the data, and then you need to calculate my average here. For the dark, I did 3 trials, looked at 3 slides. I got 65% open, 58% open, and 50. Okay. Again, if you're having trouble, just pause, and I went over those as well. You can pause it right here so that you can get your numbers, all right, and then calculate your average. So as you can see too, all right, so you already created your hypothesis. If I look over here, I can show you that typically stomata with different light usually open. So here's actually a graph from a study that was done, and they're showing that in dark, stomata usually close because it is doing the dark reactions of the photosynthetic equations and maybe doing some cellular respiration. But during white light, it is usually open to maintain homeostasis, to get in the carbon dioxide, to also take in water vapor or release water vapor, and to release oxygen if it has extra oxygen. So you can see typically with more light, they are more open. Okay, so they did actually prove this, and our data actually followed it quite simply, making sure you calculate your average there. All right, so did the data support our hypothesis? We would say yes, because the average for light was higher than for the dark, for the number of open stomata. So what is the purpose of the stomata? Again, it's to open and close, and we just need to know it's for water balance and gas exchange. How does the plant regulate the opening and closing? So again, this structure is the guard cells. Explain why a plant would typically have their stomata open during the day to do photosynthesis. The stomata of plants regulate gas exchange. What are the 2 gases? So again, they take in carbon dioxide, and the other gas, okay, is oxygen. I'm sorry. The other gas would be water, okay? And which gas is necessary for the survival? So the stomata regulate gas exchange. What are these 2 gases? Is carbon dioxide, water. You can actually also say oxygen too, because they do actually release some oxygen, but for the most part, it is carbon dioxide and water in the form of gas. G for gas. Which gas is necessary for the survival of the plant? So plants obviously do need CO2 for them to photosynthesize. Name the process in plants that need this gas. That would be photosynthesis, which you have a test on Thursday on Castle Learning for part A, and then your OneNote for part B. What organ could be homologous? Remember, homo means the same to a stomata in plants. So now we're comparing plants to animals. And how we maintain homeostasis through would be kind of like extra water. What organ? I would say would be like the bladder, because part of the urinary system, the bladder helps to release extra water that we take in. So that's just, again, a comparison. So we'll be going over some of the cellular respiration constructive response for our virtual class today. All right. So Real quickly, I'm going to go through these questions with you, and then you'll be able to calculate everything and get through. So number one, it's asking you which trait was the earliest to appear among plants. So when you're looking at this diagram, you want to see which trait is found in most of them, because that means that they obviously had a common ancestor, they share similar characteristics, and if you're looking by the columns here, the stomata is present in 1, 2, 3, 4 of the 5. So my answer is number 3, stomata. Okay, so number 1 is 3. Number 2, a student placed a test tube containing Alodia. I used to be able to buy Alodia at the fish store. I could even get it off Amazon. It is considered invasive, so actually now we're not allowed to purchase it or sell it because if people took their tanks and dumped it in the waterways, it could then spread and actually displace native species. But Alodia is an aquatic plant, and they're saying that they placed it in light, and then they calculated using the data table the number of bubbles produced per minute. Sounds like a super boring job. You sit there and count how many bubbles and multiple trials. And as you can see, the further away it was from the light, the less number of bubbles it produced. So then they're asking you, what is the gas produced? Well, obviously, photosynthesis makes oxygen, and it does it during, you guessed it, photosynthesis, so my answer would be number 4. Right. So while looking at the lower part of the leaf with a compound microscope, which we just did, you can actually notice the cells with the stomata. And again, the two things are water balance and gas exchange. That helps doing that. But they're saying, what's the main purpose of these openings? Well, we know that they take in carbon dioxide and water to make maroon 5 sugar, right? C6H12O6, because then they use that for cellular respiration. So the whole point of photosynthesis is to take in. So they're saying here, a student notices a pair of cells with openings between them. The main purpose of these openings in cells surrounds them is to, again, this one is regulating gas exchange. The second one is saying a cell in the leaf of a corn plant contains chloroplast, so number 3 is 3. And the chloroplast, again, that's what's doing photosynthesis to make maroon 5 sugar. So number 4 is 1, right? So the more chloroplast, the more photosynthesis, the more sugar. You think of the guard cells in stomata, it is water balance and gas exchange. All right, so number 5, you should be able to look at these 2 equations, and I want you to write right on your lab what equation is which. So oxygen, glucose, carbon dioxide, and water, and X is actually my, okay, taking in oxygen, this one's my cellular respiration. Remember, X would be that 38 ATP, and then carbon dioxide, water yields oxygen and glucose. This would be my photosynthesis. And remember that plants are super awesome. They have both a chloroplast and a mitochondria so that they can do both. This one will be on your test, and you will need to know plants are amazing. They can do both. We can only do cellular respiration. We are heterotrophs, but plants are autotrophs. Therefore, they have the processes to auto self-produce their own food. Okay? All right, so this next question would be a 3-point question on your regents, and you need to answer it in 3 bullets. Our next, when you guys come back, we will be starting our body systems, okay? And it's all about maintaining homeostasis in organisms. The first one is to identify a feedback mechanism or a feedback loop that is in the human body. So how do we maintain homeostasis? Well, the first thing is we could have said we maintain, well, when we come in every day, we have to have our temperatures taken, because if we are not maintaining a certain temperature, that indicates, okay, something. So that's one that we could use, okay? It would indicate that we could be sick and have a disruptancy in our homeostasis. So then we would be sent home. So our first bullet, you could say, maintaining body temp, okay? You could also say maintaining your blood glucose levels or hormones, Okay, I just chose maintaining body temperature. Second bullet is to identify, other than death, one specific result of homeostasis if it fails. The two Ds, disease or death. So if you cannot maintain your body temperature, you must have a disease or you must be sick. There's got to be something going on. You might have an infection, you might have a viral, you might have seasonal allergies. There's a lot of different things that could be indicated. If you put for the first one maintaining blood glucose levels, the second bullet you could have said diabetes. People that are diabetic can't maintain it. So again, keeping it simple. Make sure when you see 3 bullets, you answer the 3 bullets properly. Describe how plants regulate water. So the state loves to have you talk about humans' body systems and then compare it to plants, okay? They always have these type of questions. And we would say, again, my little micro teaching hand gestures, we would say describe how plants, it's through, right? Stomata and guard cells, right? Regulate the two things. Gas exchange and water loss. Okay? Bullet point maintains body temperature. Bullet point disease. Bullet point stomata or stoma, regulate guard cell, regulate gas exchange and water loss. Okay? So making sure that we can answer those. Almost done, guys. Home stretch. Number 7, again, looking at it, I tried giving you all the stomata questions so you can see how important plants maintain their homeostasis. Again, we know they open and close for gas exchange and water loss. So number 7 would be 1, okay? The change in the size of the stoma to regulate water loss. Okay. Number 8 was what I actually looked at under the microscope today for you and gave your numbers. So this is the night. You can see all of the stoma are actually closed, and then during the day they were open, which was our hypothesis, and we actually got numbers to calculate. And again, they do this to maintain homeostasis. So my answer is number 1. Number 9, some sea slugs store chloroplasts containing from algae that they've ingested. The chloroplasts continue to carry out photosynthesis within the slugs. What advantage would this activity be in these sea slugs? So I wanted just to show you for a quick second, your photosynthetic sea slugs that actually look just like leaves, but they are a slug. They have over time evolved to have chloroplasts and to be green from natural selection and mutations, and now they actually can make little babies of little baby chloroplasts, sea slugs that also can photosynthesize. So not only can plants photosynthesize, but there are some animals and microbes that can too. If they have a chloroplast, they can do photosynthesis. They are then considered an autotroph because they can make the root 5 sugar. So going back to this question, we would have to say that it would be number 1. The sea slugs with chloroplasts can synthesize, which means to make some of their own food. Okay, so number 9. All right. Number 10, same question, just a little bit different diagram for the purpose of the guard cells and stomata, water loss, gas exchange. So again, number 3 for number 10. Almost there. Plants are green because they contain chlorophyll. Remember, chlorophyll is the pigment. Chloroplast is the organelle that does photosynthesis. A bucket was left in the lawn for 1 week. When the bucket was removed, okay, the green turned yellowish and white. And actually, this is a good example too. So these plants I had in the light, you can see, still have a pretty green leaf to them. Okay, and the ones that I started to have in the dark is actually, they're starting to yellow a little bit. And that's because environmental factors can influence gene expression. But obviously what's influencing them is the sunlight. The sun is non-living, so we consider the sun A. You put A in front of something, it is not. Biotic means living, so the amount of sunlight isn't a biotic factor. Number 2, we have one more, and then my video is over, and then you guys will be able to finish and synthesize this lab and turn it in. Okay, looking back at our equations. So they're saying a biological process that occurs in plants. It's taking in water, so we have to assume that this is photosynthesis. So let's do the balanced equation here. So they're starting with water first, so we would actually, I'm just gonna write on top, they're gonna start with 6H2O plus 6CO2 yields with enzymes and sunlight, rune 5, right? It can also make, okay, so it's taking in water and carbon dioxide, and it is making, what is it gonna make? It's going to make C6H12O6 plus 6 oxygen. So we want to look at what could A possibly be. So A would have to be carbon dioxide, right? B could be my glucose, okay? C would be my oxygen, and D would be enzymes. Every chemical reactions need enzymes, so my answer for number 12 is 2. All right, so make sure this lab is fully completed and turned in by Friday. You have an exam on Thursday, and I hope to see you guys soon.